listening at me. Okay, wonderful. Okay, good afternoon, everyone. Chris Williams, and I'm here to talk about a project that Tim Long and I are working on, which is called Tissue Scaffold Fabrication via Micro-Stereolithography. So this is a 3D printing process, actually, if you will. Working with us is a master's student named Phil Lambert in mechanical engineering, and also a PhD student, Nancy Zhang, in Tim Long's lab. And it's a really nice collaborative effort to really address some pressing issues. I think one of the things that's unique about Virginia Tech is to have both a photochemistry person next door to a 3D printing person to really make some big changes. Our primary research objective is actually to build a new system, a new 3D printing process called Micro-Stereolithography, with the specific intent to create biocompatible polymers for that we can create three-dimensional tissue scaffolds with microscale features. So very quickly here, you all know what tissue scaffolds are, but what you probably know is that the ways in which they're made result in these stochastic kind of foams. There's no control over the mesostructure or microstructure of the scaffold. And we also know from research and literature that cells like specific types of scaffold structures. So as a designer, design brain, I'm thinking, well, if I know what types of structures that cells enjoy, then I should be able to design a structure and manufacture a structure that's ideal for certain tissue and cell response. So people have looked to additive manufacturing or three-dimensional printing to create ordered scaffolds for specific applications, but they tend to be like this, periodic, repeating, very limited, this sort of honeycomb sort of alternating layers. We call them log cabin structures. We've done some of that here at Virginia Tech in prior work, but that still doesn't give us a lot of options, design freedom, right, in all three dimensions. So that's why we're looking to a technology called micro-stereolithography. And this has actually been in the literature. There's a few other research labs in the US that have looked into this. So our intellectual property is not necessarily in the systems design. It comes in actually the materials development. The way in which the system works is it's like lithography, but in 3D. So we have a UV light source, that's number 2. So we have a CAD model, that's at number 1. We give a light source that goes through, that light is conditioned to be collimated and ready to be operated with. That goes through some mirrors and then bounced onto what's called a digital micromirror device. It's basically that chip that's inside that projector. It's a small array of micromirrors that you turn on and off, and that will project an image, a black and white image, off of it. And it's called a dynamic mask. So if you think about lithography, I have to create a mask, then etch, and then remove the mask. Here, I'm just creating a mask dynamically by just feeding it literally a PowerPoint slide. So, here's different layers that we can print. So off of that DMD, we then, instead of a projector, which has a diverging lens to make the image really big, we have a converging lens that makes things really, really small. So then we print that onto an elevator, and in that vat, that blue liquid is the photopolymer. So we project on the image, we cure one layer, we lower the elevator, cure the next image, and we continue layer by layer from the bottom up to allow us to create 3D structures. So if you can imagine, I just repeat those 4 images over and over again, we can create some really small three-dimensional objects. And again, that's an image from a different research lab from Illinois. So why should we do this? It's because it's really one of the very few three-dimensional printing technologies that allow us to do micro-scale features. Right now at Virginia Tech, our most high-resolution printer can do about 500 microns. That's as best as we can get. And we have no control over materials. It's not biocompatible. This will allow us to get the size scale we want and in the materials that we can work with. So in theory, once we have this process, we'll be able to actually start designing and manufacturing scaffolds for all the researchers in the nanobio realm who want to study cell response in 3 dimensions. So what we're targeting at is about a 20 micron resolution. When we first wrote the proposal, this is where we were actually. We had, with a simple projector and a magnifying lens from the dollar store, we were able to make some really small structures with an open source known photopolymer. And that's not I joked with my student, never use a Cheez-It as a size scale. Anyhow, he learned. It was the first week, actually, of him being on with us. So the research tasks were, you never know, it could be a giant Cheez-It. First task was to actually design and develop that system. Then, of course, with Tim, that's our responsibility. Tim's lab with Nancy was to actually start working on a biocompatible photocurable resin. and then characterize those properties. Then from there, we can start relating materials to process parameters, and then start making scaffolds. What we've done since the beginning of August are those first two pieces, and we're actually, probably next week, we'll have the first sets of scaffolds created. And the rest of the next year will be all about the photochemistry and process optimization. So really what Phil's work, the master student who's been working with me, has really been focusing on making these design decisions. So what types of light source, what types of conditioning optics that are necessary, what types of DMD to choose from. So, you all of a sudden get really knowledgeable about, home projection systems. So 1080p, it's not just widescreen TVs, it's 3D printing now. And then, of course, all the focusing optics about how to focus that down and then actually getting a linear stage that has exactly the resolution you need, because now we're talking about step sizes on the order of 5 microns per layer thickness. So basically, here's the details of that, again, and how this all works. Again, these are the actual micro mirrors. The mirrors themselves are 10 microns, so it's just equivalent to a pixel on your bitmap. You turn that on and off, and I can which means they just move the mirror in or out of the light path. And that allows me to dynamically generate masks. And we can reduce that projected image quite a bit with some very small feature sizes. So Phil, I guess it was about maybe a month or two ago, had his CAD finalized, all of his optics designed and selected, and now it has come to life. So what you have there is the UV light source. It's an LED base, goes through the beam expansion and light collimation, bounces off. I don't have a mouse. Here we go. I have this thing. Sorry. So it goes through here, bounces off the mirror, then up to this DMD mount. The mount itself is a gimbaled system, and it's all been 3D printed, of course. And then that goes down into the focusing optics and into the resin vat. So let's see. So our next steps, we've got to finalize the assembly. We're waiting to finish the design of the actual motor mount. And then he's just completed some control algorithms from LabVIEW. And then, of course, comes the science, the real, step away from engineering, maybe more to the science of relating the materials back to the process itself. This is work from Tim's lab. And so now this is where I start fumbling. The bottom line is what Tim has done and his lab has done is generated a biocompatible photocurable resin and has functionalized it to reduce the viscosity, which makes the processing much quicker and much easier for us, because as we lower the elevator, the resin level has to settle, and by having lower viscosity, that will make that process a lot faster, so it should improve printing time. And again, the key thing, of course, is getting something that's biocompatible, something that cells will survive on. So with that, basically the highlights here are that we finalized our system design. We're completing our machine assembly by probably tomorrow, and we'll start curing resin selectively actually next week and the week after. So we have planned NSF and NIH proposals upcoming, and we've already got a publication drafting on the design of the system for the 3D printing world. The big successes actually are that we've actually engaged a mechanical engineering senior design team to assist with this design. They're making a smaller scale system that's portable that we can take to high schools for outreach kind of activities, as well as do some other materials development. And we've also worked on material science to help characterizing that polymer. And we've got some other cool things going on. One of our key collaborations is we still have ongoing talks with a 3D printing company to look at sort of integrating some of the biocompatible polymers into their manufacturing platform. So with that, I'll stop and answer questions.
We might save those for the next break, okay, so that we can stay on track here with Tiana. Thanks, Chris.
Can I ask a question while we are switching?
Sure.
So I think that the technology is really interesting to control the geometry of the scaffold, but at a pretty large scale. How would you take care of the viscosity of like elastic properties of the linear? Because it's not only geometry of the forge, but cells also sense elasticity of the linear.
Yeah, so you would actually print the scaffold and then take it out of that suspension and actually then pass it on to you to then do all the traditional.
Oh, to put some things in.
That's
okay.
right. So the cells will not be in that resin. We won't be selectively curing the cells. They will be utilized probably.
Okay.
Yeah.
Okay. So, it's me again. So, I'll tell you now about the project that is actually a collaborative project between my lab and Paul Moore's lab. And it has a this is the short title, modular biotemplates. This is a long title. So, the two students in my lab working now full-time on this project are Nathan Carter and Shigen. And this is the big picture of the project, but then I'm going to split it into pieces because it's modular. So what we are really interested in is to combine biological self-assembly, molecular recognition for a purpose of templating of nanoparticles. And what we are really interested in are not individual nanoparticles, but collective properties of the ensemble of nanoparticles. And so the idea and what we think is unique in this proposal is that through this biological molecular recognition and self-assembly that is happening really on the nanometer scale, we have a possibility to pattern this nanoparticle array with a single nanoparticle resolution. And so this is going to give you much better resolution than the top-down approaches that are currently used. And so the strategy is depicted here in this little picture, where in my lab we design these are de novo protein sequences that have self-assembly properties. Within these protein sequences, we can engineer specific peptide binding sites. On the other hand, we can take the nanoparticles, we will modify them with the peptides that specifically bind to these proteins. And now when we mix nanoparticles and proteins and assemble thin films out of this material, depending on orientation of the peptide binding site within the self-assembly protein, we can make a thin film in which nanoparticles will array in different geometries. And our hypothesis is that depending on the geometric arrangement and orientation of nanoparticles in respect to each other, these thin films will now have different collective properties that we can see in their absorption properties and light properties. So this is the protein design part of it, and this is what we consider, why we consider the system to be modular. So we design modules that are binding peptides, and then separately design so-called spacer modules. And through appropriate combination of the B modules and S modules on a gene level through recursive ligation, we now can make the superhelical proteins that will have a peptide binding sites with different kind of orientations. And the first generation of the proteins that we made that we're looking here are these superhelical proteins. They're about 18 nanometers long, and they have a peptide binding site 120 degrees offset from each other along the superhelical